Hey everybody, I just wanted to take this as an opportunity to kind of talk to you about our lab from this last week. What we were doing was we were trying to do a plasmid extraction. We used a mini prep kit, and what we were really trying to do was get, if you take this as a E. coli cell, I'm going to use a couple of different colors here. I'm going to use green to show my genomic DNA. And I'm going to make sure that I bring that back to make sure that it is a circular chromosome, but it's all tangled up because what that DNA is going to do is it's going to coil in on itself. It's going to kind of, it's a big circle, but it's going to like collapse in on itself. Um, then I'm going to use purple to show our plasmid, which is right here. Our plasmid is about 1,000 times smaller than genomic DNA. It doesn't have any it is not essential, but what we can do is we can basically put pressure on our bacteria to hold on to that plasmid. And so we use antibiotic resistance genes on our plasmid. AMPR is sometimes what you'll see for the ampicillin resistance gene. And that is basically going to say, let's go ahead and I'll give you two different examples. Here is an E. coli cell, and I'm going to make sure to draw the genomic DNA. Here's the genomic DNA. One of these two cells, A and B, one of these two cells is going to be killed by the antibiotic or in the presence of the antibiotic ampicillin. And that is correct. It is B. B will be killed because our plasmid has that ampicillin resistance gene. So it's going to produce the enzyme that will basically render ampicillin ineffective. So this will be safe from ampicillin. Um, Now, with that in mind, that's basically what we're trying to do. That's what we're trying to select for bacteria that have that. And then what we're trying to do is purify that plasmid. And so we worked through the plasmid purification and we burst the cells open. We did a chemical um, plasmid extraction, which I'm just going to label it as plasmid. Well, I'll label this as PE. So plasmid extraction. So we did a chemical PE. Some of the things that were involved in this chemical plasmid extraction was the enzyme RNase, because what that RNase is going to do, it's going to degrade up any RNA because, yeah, absolutely, there are going to be genes that are transcribed that, these bacterial cells are going to undergo or they'll be actively doing transcription and translation. So RNA will be produced. Our RNase will knock that out. So that RNase will destroy RNA. Then what we'll also be doing is this chemical plasmid extraction will have a basic or an alkali, an alkaline environment. I'll say environment, and then I'll also put in parentheses here pH. And what that's effectively going to do is not necessarily destroy, but it's going to really disrupt our genomic DNA. Because what we care about is the plasmid. All that we want to recover is the plasmid. We don't care about the genomic DNA. And so whenever we're breaking our cells open, if we have something that can disrupt genomic DNA, then we want to take advantage of that. And that basic environment, that high pH environment, will destroy the genomic DNA. So our chemical plasmid extraction will do all these things. RNase will destroy the RNA, and our basic environment will disrupt the genomic DNA. Now, whenever we're all said and done with this purification, we were collecting we used a nanodrop. And with that nanodrop, we looked at the absorbance at 260, 230, and 280 nanometers. Now, these wavelengths are all used for different things. In particular, whenever you measure wavelength at about 260, 280, these two, effectively what you're able to do is determine the purity of your sample, because 260, 280, we can use that for a determination of protein or DNA to protein. Because on page 20 of your lab manual, if you start at the top, first paragraph, second paragraph has an explanation of the Lambert-Beer law, and then this third paragraph, third paragraph says, for best accuracy, absorbance of a substance is measured at a wavelength of maximal peak absorbance. In the case of nucleic acids, DNA and RNA, the maximal absorbance is 260 nanometers. So 260 is DNA plus RNA. For DNA/RNA, the measurement is independent of the composition of the molecule. So that doesn't matter. Or basically what that says is it could be it could be nothing but A's. It could be nothing but T's. It could be nothing but AT. It could be a 100% AT content molecule. It does not matter. The sequence is not going to influence this. Proteins maximally have an absorbance at 280 nanometers. So this is protein. So we look at a 260 to 280 ratio. And then last but not least, we're also looking at 340 nanometers because 260, 230, that's a ratio that we look at that basically tells us if we have any sort of contamination or chemical contamination from the kit, the mini prep kit. Now, what we're most interested in is the 260, 280, and what is like ideal is a ratio that's approximately 1.8 to 2.0. Most of the results that I saw were about 1.77, 1.75. I would argue that that is pretty much close enough in the sense that it's 1.77, 1.79. That's effectively 1.8. I would say that the folks who got something that was like 1.0 or 0.47, well, that's going to be that just means that you have a ton of protein in there. How much that's going to impact your results, well, it remains to be seen whenever we digest that DNA, but that's kind of where we are. And so, it looked like everyone did a really good job from what I saw, and so keep it up. The last thing about this was those calculations that were on page number 23, and it seemed like everyone got through those calculations without too much of an issue. And I think in large part that's because most people's concentrations of DNA were about, I'm going to say 120 to 280 nanograms per microliter. That was the range that I saw. There were some that were considerably lower. There were some that were considerably higher. But that's what I basically saw, and those are good numbers. Now that's helpful because what we'll be using this for in the future is a part of that calculation on page 23, and that was, well, what volume is needed to get 250 nanograms of DNA? And so if we were to take something like this, 120, if you had 120 nanograms per microliter, well, I always like to think about this and I always like to say, okay, let's imagine I have 1 microliter of this solution. What mass of DNA do I have? I have 120 nanograms. Okay. So how many microliters do I need to have 250 nanograms? Well, let's see. 120 is a little bit less than half of 250. So 2 microliters of this solution would give me approximately 250. It's going to give me 240 nanograms. I mean, it's important to do the calculation, but I think that it's helpful to look at it in that sense. Okay, well, I hope this is helpful, and I hope that you have a great week, and I will see you in lab this week.